In this video, I'm going to talk about the clustering module. The first thing I want to draw your attention to are the menus on the left-hand side. From these menus, you can choose, first of all, whether you want to consider all the genes or just a subselection of them based on these features. You can choose whether you want to leave your samples ungrouped or whether you want to group them by phenotype. And you can also decide whether you want to focus on a specific set of samples. This can be useful in the case you have a very large data set. Finally, if you click on options, you can decide whether you want to perform your analysis at the gene or the gene set level, and whether you want to exclude genes in the X and Y chromosomes. The clustering module, first of all, generates a heatmap that shows the clustering of your samples. If you click on the settings icon at the top, you can choose whether you want a static heatmap or an interactive heatmap. And with the interactive heatmap, you can then click on single blocks and get more information about the gene and the samples they come from. You can further customize the heatmap by splitting the samples by phenotype or gene, as well as selecting one of these options. So, for example, by default, the heatmap will only display the top 50 genes and 4 clusters, but you can change these values here and here. You can also download the heatmap as well as any other figure that you will see on the platform via this icon. You can download it either as a PNG file or as a PDF file. And in the new version of the platform, you can also choose the size of the PDF file. The next tab shows you a scatter plot with 3 different methods, PCA, tSNE, and UMAP, that shows a clustering of your samples in the dataset. Now, if you go to the settings icon, we can change the color and labeling of the samples by phenotype. So in this case, I'm sticking with the glucocorticoid phenotype. We can also change the shape of the dots according to one of the phenotypes. So in this case, I'm going to go for treatment. And now, if we look at this particular data, you can see that while the samples are neatly separated into resistant and sensitive, there is also a subdivision between treated and untreated samples within each of the 2 clusters. If you want, you can label each of the individual samples, and you can also visualize the plots as 3D plots, which are furthermore interactive. Going back to the 2D version of the data, we can also choose which of the methods we want to use to cluster the data. The default version is a tSNE plot, but you can also choose a PCA or a UMAP plot. And finally, you can choose the number of genes that you want to reduce to use for dimensionality reduction. Next, we have the Parallel tab. In this tab, you can see a parallel coordinates plot that displays the expression level of selected genes across all conditions. This plot is particularly useful if you're looking at a time series. So in this case, it makes sense to group your samples by phenotype. I'm going to group them here by cluster, but you could imagine this could be by time. And then you have the expression of selected genes as they change across time. And you can then order the various conditions along, because this is an interactive plot. So you can order them chronologically and then have a neat picture of your expression for these genes over time. And here at the bottom, you can then look at all the selected genes that are represented here, and you can see their average levels of expression across all conditions. Finally, on the left-hand side, we have 3 more tabs. The Annotate Clusters tab relates to the clusters produced by the Clustered Heatmap tab. And here you can see the 4 clusters with their functional annotation. You can further customize this plot by selecting the reference level, which can be at the gene set of phenotype, with the gene set being the default choice. And if you stick to the gene set, you can also choose which reference set to use. Default choice is hallmark collection, but there are many more, including KEGG metabolic pathways and goal terms. You can also click an option here that will add a Fisher test weighting. At the bottom here, you can see a correlation of the different clusters with the, in this case, hallmark gene sets. Next, we have the Phenotype tab that shows you basically tSNE plots colored by the various phenotypes present in your dataset. And you can customize it by deciding whether you want to put a label with the samples or as a legend. And finally, we have the feature set ranking plot, which shows the rank discriminant score for the top feature sets. And again, you can customize this by choosing which method you want to use to produce this plot.